Hello, everybody. Okay. Let's see. I think I'm sharing the wrong thing. Hang on. I want to share the whole desktop. Okay. Oh. Okay. There we go. All right. And then I also want to do our iCloud. Where did it go? Here we go. All right, everybody doing good? Staying out of the wind at least. Video is not showing. 7.2 is not showing in YouTube. Let me see. Let me see. All right, let's see. Hold on. I have two screens, so when I look away at the other screen, that's what's going on. All right. 7.2 is in October. Wait, what? 7.2. Oh, yeah. 7.2 we did in class. 7.2 we actually did in class. That was the lecture. Just like today, we're doing 7.4. But it is recorded because it's a Zoom of class. It's in the Zoom thing. So watch. If you go to YouTube, I know if you go, let me do it the way you have to do it. So if you go through Moodle, go to our class, and then on the class here, you have this link, Chem 435 4M Zoom class. Oh, no, I take it back. That's not right. Where did I put that? Here, link to Zoom recordings, right? This is the link to the pre-recorded. This is the link to the Zoom. I'm sorry, Hannah. Okay, so then, where are we? That's August 4th. Was it when? No, it's in here. September, it was October. Where's the October? Did I put it in the wrong place? Where did y'all find it? Yeah, I don't see it either. This is really weird because I know I uploaded it. Annie, you couldn't find it. Did I not upload it? Or did I upload it and not put it in the thing? Hang on. There are so many videos. It is hard to manage. Okay, let's see. My videos. You just have to look in your general video category and not in the playlist. So I didn't put it in the playlist. Let me see. Here, Wednesday, October 7th. So wait, let me see where it is. And it needs to go in the right playlist. See, I didn't put it in a playlist. I'm going to put it in the Zoom of class because that's what it was. Here we go. So that's my fault. It wasn't in the playlist like it was supposed to be. Save. Okay, so now if I go back to my playlist, here we go, playlists. It should be Zoom. It should be in here. And I'm gonna have to move it up. Yep, and I'll put it to the top of the playlist. Y'all always please tell me if you can't find something like that. And I will fix it. But sometimes I just don't know that that's going on. I do my best to get them up as soon as possible. And sometimes when I'm trying to get them online, I forget to put them in the right playlist or something like that. So y'all, please tell me. Please tell me. Okay. Yay. Okay. And if you're looking for the lab video, if you've, it's not follow me. What's the right term? Subscribe to my YouTube channel. That's so weird and awkward. But, right, if you go back, and I'm not very good at getting around in YouTube, so you'll have to forgive me. I have another playlist that says Chem 437 Fall. Remember to like and subscribe. That's so crazy. But the video's there. It's me. I think he's just going to use the video that I made from the lab for Thursday's lab class as well. So you can find the video here. Okay. Anybody else have any questions before we start? My people who are on Friday lab, were y'all able to find the information for lab that was supposed to be today? Hannah, I'll never take it down, but don't tell Dr. Smith. So my people who are in 437, can y'all check in real quick on the chat and tell me if you were able to find in Moodle all of the information you need for lab today? Please check in on the chat and tell me yes, no, maybe so. Because if you go to lipid purification, right here, lab 4 material for video lab, right? There's the lecture material. There's the link to the experiment, and these are my 2 errors. So make sure you notice that. And then the data. Okay. All right. So we're okay on that? Yay. All right. And I was trying to do, so what do we do about the errors? You don't have to do anything. I just say it wrong. It's right on all of your stuff. I don't always say things correctly. What can I say? It happens. But it's all right on all of your papers. So let me log into iClicker Cloud, and then everybody can put in their attendance. So you get credit for being here, being troopers on a hurricane day and being here. It's so awesome. Okay, Nichols. I'm sorry, I got sidetracked on the YouTube thing. So let me start class. Okay. And then, I'm sorry, I got sidetracked, so I should have been already started. But I have a short lecture for us today. It's not long. It's really not long. And so I think you're going to like that. Yay. Okay. Here we go. 4:35. And we'll do attendance real quick. Let me bring this over. Here we go. Okay, so go ahead and check in for attendance. I'll give everybody a second. Okay, so I'll leave that running. I'll leave that running for y'all. And we're gonna start talking about enzyme kinetics. Woohoo! Enzyme kinetics is really cool. Can we have a joke? Oh, I think I forgot to get a joke ready for y'all today. So let's see. I have to come up with a good one. Hang on, watch, hang on, wait. Okay, this is a good one for me. This is right on for me. What does a cow like to drink before bed? Decaffeinated coffee. I love my coffee. I love my coffee. I have some here, all right? So yes, thank you, thank you, thank you. Another bad joke from Dr. B. I have a plethora of them. We could do this forever. Okay, so on to some actual science, right? We actually have to do a little bit of science today. So we are talking about enzyme kinetics. So enzyme kinetics is really cool stuff. It's really, really important that we can determine mechanisms for enzymes, right? So if we know the mechanism by which a reaction takes place, we can learn a lot about that reaction. We can learn about the effects of regulatory mechanisms on all kinds of different enzyme kinetics. We can look at how changes in reaction conditions are going to affect a reaction. And so enzyme kinetics is the quantitative, like we can actually put numbers to it, study of the rate of chemical reactions that are performed by enzymes. So it gives us a lot of information. And we want to be able to relate a reaction rate to free energy, right? What's our symbol for free energy? Delta G, right? And we also want to be able to relate that to equilibrium. What's your equilibrium symbol? Y'all can actually say things on the chat. I'm monitoring the chat. Anybody? Equilibrium? Yeah, I want y'all joke, but I'm gonna hold out on my joke until I get some responses on the chat. Equilibrium is K. Y'all remember that? And y'all know there's a difference between big K and little k, right? And we're going to talk about that today. Do y'all remember this from chemistry? Did y'all go over a little bit of kinetics? Yes, yes, big K, yay, yes. So are y'all typing really quickly and it's just my internet that's slow? I don't want to be, no. Y'all crack me up. Y'all crack me up. Y'all should have just blamed it on the internet. All right, so we know enzymes. We know enzymes speed up reactions, right? My voice is going out a little. I don't know. This is the only internet I have at my house. I might have to do this as recorded if we have to go. Oh, so maybe it's your internet, Taylor. Okay. Well, it'll be recorded. If anybody has trouble seeing this, it'll be recorded and you can skip ahead about 12 minutes into the video. All right, so we know enzymes speed up chemical reactions by doing what? What do enzymes do? They lower the activation energy. And there's another way to write activation energy as delta G double dagger, right? So they're going to speed up that activation energy. But remember, yes, remember our delta G is unchanged, right? Unchanged, because that's a state function, right? So today we're going to see, which, what other things affect our enzyme kinetics. So velocity is going to be one of them, enzyme velocity, and substrate concentration. So we're going to see how these two things relate to our activation energy. So now that we can use this quantitative ability to understand how enzyme activity relates to protein function, we can define what's called, at the end of today, we will be able to define what's called the catalytic, C-A-T-A-L-Y-T-I-C, catalytic efficiency of any enzyme. That's our goal for today. It's a great goal. And if we understand the catalytic efficiency, this is today's goal, then we understand its biological effect inside of the cell. So you know, I hope you know from previous chemistry, K, little k is our rate constant for a reaction, right? So dredge back up that Chem 2 information. So our velocity, this is one of the new things we're talking about, is our velocity of our reaction is a combination of two things. It's a combination of the rate constant of the reaction and the concentration of our substrate. So reaching back to that Chem 2, which reactions, we're talking about orders of reactions, depend on the initial substrate concentration? Zero order, first order, second order? Anybody have ideas? First order, yay! First order, because, right, our initial substrate concentration controls the reaction rate. So this is kind of a condensed version of the equation we were talking about Wednesday, right? Where we said our enzyme plus our substrate goes to an enzyme substrate. And then we talked about that enzyme substrate going into an enzyme product, and then that enzyme product separating and the product being released. So what we're going to show is how we eventually get down to a little more reduced formula, and how we kind of simplify that, because that was kind of complex, right? Just a little bit. So when we're able to simplify things, we're going to use what are called Michaelis-Menten kinetics. So Michaelis-Menten kinetics are only for first order reactions, right? And remember, when you're talking about that rate constant, when we talk about here, this little k, what are the units on little k? Anybody remember? Little k? And I know my little k looks like an R, I'm sorry, but my little k's always look like an R. Seconds to the -1, that's my units on little k. So if we talk about velocity, right? If we know velocity is seconds to the -1, what are my units for substrate? So if we have seconds to the -1 for little k, then my substrate would be what? Big M, molarity, right? So our velocity units are going to be, you can rearrange them like molarity times seconds to the -1, right? So we can do that. Now, this does not work for a second order reaction, right? Because for a second order reaction, we would have another product. We would have like your enzyme, you would have like an enzyme plus a substrate plus another substance. And so our velocity would then be little k, and then you would have two reactants that it would be dependent upon. So we can't do this. We can't do it with second order. So we're not going to worry about second order reactions. You can kind of just cross those off the list, which is really nice. But when we're measuring rates of reactions, how do we measure that? What are we gonna measure? If we want to measure a rate of a reaction, I guess I should kind of add in, because I'm feeling like y'all need a little extra on this. How do we measure a rate of a reaction? Right? Experimentally, enzyme, if you have enzyme plus substrate going to product plus enzyme, what are you going to measure? There are two things that you can measure. Change in concentration over time, that's exactly right. Of what? You could either, very nice, you could either look at loss of substrate, so loss of substrate, or gain of product. And a lot of times what we do, we talked about this in lab, is we measure gain of product, because you're starting out basically at 0. And so it's easy to see small change. Whereas when you look at that that substrate, right, it's really, really high. And so to see a small change for it to go down is going to be really actually pretty difficult to measure in lab. So we tend to look at the product going up, right? And so if we do that, right, if we graph, if I could write, if we graph our concentration of product versus time, right, what should we see? We should see something that increases over time, right? Now, how is this graph going to look different if I increase my substrate concentration? Anybody? It'll be steeper. Very nice. It'll be steeper. So it'll look more like this, right? So as we go this way, we increase our concentration of our substrate. That's exactly right. So as we increase the concentration of our substrate, the rate of the reaction is going to go up. So if I increase it some more, it's going to increase. Oh, that's a bad. We're going to increase some more, right? And how about if we keep increasing, what's going to happen? Anybody see what's happening? We're going to make more product, but eventually increasing our substrate isn't going to make an effect on our rate anymore. We're going to get to a maximum rate. Like, it doesn't matter how much substrate you have, your enzyme is sitting there and you've gotten so much substrate that you have filled that enzyme's binding pockets. All the enzyme's binding pockets are saturated, and you can't make that reaction go any faster just by adding more substrate. So you get to a maximum velocity. So you're going to reach a maximum velocity of the reaction at some substrate concentration. But what do you notice about that change? When I added a little bit in the beginning, when I went from here to here, I got a huge change. But the closer and closer I get to that maximum, the smaller effect that increase in substrate is going to have on the velocity of the reaction, right? So as we increase our substrate, you're going to reach a point where increasing that substrate does not affect our reaction rate. And so we call this max velocity. It's a little v, Vmax. Okay, and so why do we get to Vmax? This is a saturation of binding sites on the enzyme. That makes sense? Good. Okay, so if I took this graph and for each one of these lines has a certain initial substrate concentration, right? Each one of these lines has a different initial substrate concentration. And so if I took that initial substrate concentration, can I go down? No. So if I took that initial substrate concentration and graph that versus the rate, what do you think that graph would look like? It would look hyperbolic, which means that it would be very steep in the beginning, and then we would get really close to that Vmax. We will probably never quite reach that Vmax, but we would get really close. So what that is I would have what's called the Vmax right about here. So this point right here is the Vmax, the maximum velocity of our enzyme reaction. And so this is perfectly indicative of a first order reaction. So this hyperbolic curve tells me I have a first order reaction. So this plateauing, the plateauing, well, it's not just the plateauing, a hyperbolic curve. Oh, that's okay. I know what you're talking about. I never spell anything right. A hyperbolic curve is very, very steep, and then it levels off, it plateaus. So that's the hyperbolicness. It's very rapid, and then it pitches over. Okay. All right. So I want to show you, we're going to get into more details on this, but I kind of want to give you an overall picture of the whole place we're going. And your book has this really awesome, really short little animation that I want y'all to see that kind of shows you the enzyme and the substrate moving and all that kind of stuff. And so we're going to watch this little animation, and then we're going to kind of nitpick it and understand the kinetics behind it. Okay. So let me change this. Let's see if I can change this. Let me make that smaller. Okay. And I want this. And I already pulled it up, I think here. Yay. Okay. How do I do this?
Enzyme.
Okay, wait. Let me make it big for everybody. Okay.
Enzyme kinetics. Measuring the Michaelis-Menten kinetic parameters of an enzyme is how scientists characterize and compare enzyme-catalyzed reactions. The overall enzyme reaction can be represented by the following formula, with the enzyme E and substrate S reversibly associating, reacting, and rapidly dissociating into the enzyme and product P. If we graph the concentration of enzyme substrate complex, and product over time, we would see an initial burst of product formation that corresponds directly to the first step of the reaction, enzyme substrate complex formation. This is called the pre-steady state period. After the pre-steady state period, the reaction reaches steady state, where all enzyme reaction steps are occurring at a steady rate. This is the steady state period, and during this period, the substrate concentration declines steadily as product is formed. By plotting the change in product concentration during the steady state period and finding the slope of the line, we can calculate the initial velocity of the reaction. Initial velocities are important for finding Michaelis-Menten parameters. When we determine the initial reaction velocities for different starting substrate concentrations, we see two trends. In these experiments, the enzyme concentration and the volume of the reaction are both held constant, whereas the substrate concentration is varied. First, the higher the initial substrate concentration is, the higher the initial velocity. Second, although initial velocities increase, the increase is not as great at higher substrate concentrations than at lower ones, because at high concentration, we are beginning to saturate the binding sites of the enzymes in solution. For this reason, when we plot initial velocity versus initial substrate concentration, we get a hyperbolic curve that is characteristic of first-order Michaelis-Menten kinetics. Using this plot, we can now determine other important Michaelis-Menten kinetic parameters. The velocity that the curve approaches asymptotically is the maximum possible velocity of the reaction, or Vmax. If we divide the Vmax by 2 to find half the maximum velocity, the corresponding initial substrate concentration is known as the Michaelis constant, or Km. Increasing or decreasing the enzyme concentration will change the maximum possible velocity, but will not change the Michaelis constant. It's a constant property of the enzyme reaction. Another way to look at the same data is to plot the reciprocal of the initial velocities and substrate concentration. This converts the data into straight lines and is known as the Lineweaver-Burk plot. Lineweaver-Burk plots make it easier to obtain the Michaelis-Menten parameters. The y-intercept corresponds to the reciprocal of Vmax, the x-intercept to the negative reciprocal of Km, and the slope is the ratio of Km to Vmax. Another way to compare enzymes is to determine the maximum catalytic activity under saturating levels of substrate. This value, called the turnover number, is defined as kcat. The kcat for a reaction describes the catalytic rate, but it does not reveal the catalytic efficiency. In the case of the enzyme fumarase, the Km for fumarate is 5 times lower than it is for malate, yet the kcat values for these two substrates are nearly the same. This results in a specificity constant for fumarate that is about 4 times higher for the enzyme than when malate is the substrate. This higher efficiency means that fumarate is the preferred substrate of the enzyme.
Okay, so that was a lot of information in like a really short 4-minute little animation. So what we're going to do is we're going to go over all of that stuff in detail and make sure you understand it. But it's a really good video to be able to go back to. And so I posted that little 4 minutes separately on YouTube. I think I put it in the lecture video playlist, but it's on YouTube, and I put the link on Moodle where that animation should be. So in chapter 7 stuff. Okay, so what they were talking about in the video, right, is that if here we're graphing the concentration of our substrate versus time, right, what's going to happen? Well, if we're talking about our reactant, it's going to go down with time, right? If we talk about our product is going to go up with time. But what's the difference between this pre-steady state and this steady state conditions? Both of those two things are still changing. So what's being held at steady state? Why do we call this steady state? And y'all can tell me in the chat. So what is the enzyme substrate complex? That's right. The enzyme is constant. Excuse me. So this is the important part. This is the super important part, that this enzyme substrate complex is at a constant concentration. And so when we have this steady state conditions, this is when we can graph our concentration of our substrate versus the rate of the reaction, and we can get our hyperbolic curve, and we can find our Vmax, right? Well, that's great. But using this information, we can understand a parameter about our enzyme that is going to be constant no matter what concentration substrate you have. And that's called the Km. So if we find, in order to find the Km, and they call it Km because it's the Michaelis-Mitten constant. So the Michaelis, A-E-L-I-S constant. It's a constant. So it's constant no matter what the concentration of our substrate is. So if we find our Vmax, and if we go halfway to the Vmax, so this is right here, we are at 1/2 Vmax, and you drop down, right? What you're gonna find is the Km. So you're gonna find the rate at which our product formation is going to happen. It will end up giving you a concentration of our substrate that will give you that velocity. Oh, big K. It's a big K, Ross. You can use it to find concentration of our substrate, right? That will give you half of the maximum velocity. That will give you half of the max velocity. That's the important part. Okay? Now, if we look at kind of the math behind this, right? Our Michaelis-Mitten constant can be described understanding the rates of the different reactions that occur. So remember our little equation, if we had our enzyme plus our substrate, and it was a reversible, and it would go to enzyme substrate. Now, we're kind of going to assume that going from enzyme to product, this thing right here is going to happen super fast. That's our kind of assumption in this reaction, so that we can kind of just skip writing this out. And we're also going to say that release of the product is going to happen very quickly. And so these are assumptions, but you have to make some assumptions in science sometimes to make the math doable. So we're going to get our enzyme product. This is how we kind of cut down that equation. So each of those arrows that I just drew actually has a reaction constant associated with it, right? And remember, our reaction rate is our little k. So the first one is K1. Then the reverse reaction going the opposite direction is going to be K2. Not K2, K to the minus 1, because you're going, it's still K1, but you're going in the opposite direction. And then this one is K2. Okay, so this is going to give us a way to relate our initial velocity to our Vmax, right? But it's going to depend on our concentration of our substrate. So we can write it in our formula here, right? So it's just including all of the rates of our reactions, right? But what we want to do is we want to be able to relate that to our substrate concentration, and we want to be able to relate that to Vmax. And your book goes into a derivation of all of these equations. And if that sort of thing is your deal, have at it, have fun. But I'm not expecting you to understand the mathematics behind the derivation of these equations. Please, please don't spend a lot of time focusing on that, right? But if we understand these parameters of our enzyme, if we understand these parameters of our enzymes, we can describe the enzymes and compare them and say, what's going to happen if we add an inhibitor to it and all that kind of stuff, right? So when we look at these kinetics, what we want to do is we want to be able to say, what happens if we change a parameter? Well, what happens if we decrease our amount of enzyme? So here's an example situation of something that's going to happen in the cell, right? We're going to start out with a high enzyme concentration, and for whatever reason, there are many reasons why, you would decrease your enzyme concentration, because the next section we're going to talk about how enzymes are regulated. And so this is something that happens in the cell all the time. Even if we decrease that enzyme concentration and our Vmax changes, right? We had a high Vmax for a high enzyme concentration. We have a low Vmax for a low enzyme concentration. What is not going to change is our Km value. Our Km value is a constant, right? That's never, ever, ever going to change. So even if your concentrations change, your Km should not change. And so it allows you to have a constant so that you can make calculations and understand what's happening. And we can actually relate this to our KD. So remember what your KD is. KD is your dissociation constant. T constant, right? This is really, really important in talking about an affinity of molecules, right? So your, if you assume, let's see, if your Km, right, was your little K1 plus your little K2 over K, oh, this was negative. That one's -1 over 1, right? This is our Km equation. Well, if our product formation is slow, right, that means that our K to the -1 is a whole lot bigger than K2, right? We can assume that our Km is really K to the -1 over K1. And that is actually the same thing as the KD. So we can relate this to affinity, which is awesome, because now we can measure affinity. We have a way of measuring that in a test tube. So if we have a high KD, right, what does that tell you about affinity? or a high Km, what does it tell you about affinity? A low affinity, great. Low affinity. Great, awesome, right? Now you can't always make those assumptions and make Km related to Kd, but a lot of times you can. And so it makes it a very easy way to mathematically calculate affinity of an enzyme to a substrate. So it's really nice. It's really, really nice. But sometimes these graphs are really hard to look at and to analyze. So a lot of times what they do is they linearize this data, and then they represent it as a Lineweaver-Burke equation. So this is like a graph of a graph of a graph. But it's just the double reciprocal. But it's okay, you don't have to understand like how the graph was generated. You don't have to go there. But this is just giving you all the same information, right? You can find the x-intercept is our 1 over Km, right? You still get Vmax. And then the important thing that you really want to compare is you want to be able to compare your Km versus Vmax, right? This is what's going to tell you the most about your enzyme kinetics, right? And so it's just an easier way to represent it. It's not a big deal. So, like, I don't want you to spend a whole lot of time worried about, you're never going to have to generate a graph like this or that kind of thing. You just kind of have to be able to look at them and know what information you can pull from that kind of an equation, that kind of a graph. Okay, does that make sense? And I'm watching the chat. Yeah, okay, good. All right, so that tells you about basically affinity, right? But just because something will associate, you have an enzyme associating with a substrate, you don't have to draw the graph. Yep, you don't have to draw the graph, just interpret. So you can talk about binding affinity, how tightly a substrate will interact with an enzyme, but you actually really need to understand, in addition to that, what's called catalytic efficiency, right? So if you have a saturating level of substrate, so our reaction is no longer going to be dependent upon substrate concentration, then that will give us our maximum catalytic activity. So it's called kcat. And so kcat is really the value of our rate limiting step, right? That's what kcat is. This is our rate limiting step. That's what it's determined by. Y'all did rate limiting steps, right? Y'all know what those are. No. Okay, you don't. Okay, so anytime you have a reaction, usually it's a multi-step reaction that's going to happen. It's not just A plus B goes to C, right? We write A plus B goes to C because that's the net equation. But it's always going to be in parts, and we're going to add them all up, and we're going to get that goal equation. Well, one of those steps is going to be slower than the others. And so the step that determines the overall rate of the reaction is the slowest step, is the slowest part of the reaction before you get to the whole. So like you want to get all your friends together and you want to go to Starbucks, right? One of them is going to be your rate limiting factor. One of them is going to determine what time you get to Starbucks. If everybody else is early, it doesn't matter if that one person holds you up, right? It's the same thing in an enzyme reaction, right? We have multiple steps that are going to happen, and one of those steps is going to take the longest. And so that one step that takes the longest is really going to determine the rate of that enzymatic reaction. Okay. Make sense? Yeah. Okay. So, so when we talk about our kcat, and it's a lowercase k, kcat. No, it's an uppercase k, kcat. Kcat is really the Vmax, right, over our enzyme total concentration. So our enzyme total concentration is really our enzyme plus our enzyme substrate. Okay, so you can have two enzymes and they can have the same kcat, they can have that same slowest rate step, but they catalyze reactions at a different rate because they have different Km values. Or the reverse could be true. They can have exactly the same Michaelis-Menten kinetics, the same Km value, but they catalyze reactions differently because their kcat is different. So you have to take into account those two factors when we're talking about an overall enzyme reaction, right? And so that is what gives us our catalytic efficiency. Remember I said that was kind of the goal for today? How do we get enzyme efficiency? We have to think about two things. We have to think about what's dependent upon substrate concentration and then things that are independent of substrate concentration. So independent of substrate concentration is kcat. Dependent on substrate concentration is Km. And so it's the joint combination of those two factors that really tells us our catalytic efficiency of our enzymes. Okay, so that's generally how it works. Now, you're in a cell. Cells don't always behave the same way all the time, right? They're gonna have kcat independent of what? kcat is rate limiting step independent of what? That is true. Independent of substrate concentration. Yes, good. Okay, so that's true for catalytic efficiency. But remember, our cells have changing environments, right? So what are two things that could change our enzyme kinetics? And that would be pH and temperature. pH is really cool because you have to look at what's going to change when you change a pH, right? If I change pH, what's going to happen to any protein? I'm not talking about just an enzyme, but any protein. If you change pH, what's going to happen in a protein? Protein will. There are two things that it could kind of affect. It can affect this overall structure of our enzyme, right? It's a protein. Does pH affect protein folding? Absolutely. What about the chemistry of the active site? Will change of active site. How would this change the chemistry of the active site? I'm going to wait for an answer on this one. I really am. What changes? If we change the pH, think about what happens when you change pH. Ah, awesome. Protonation and deprotonation of amino acids. Yes. We have 7 amino acids that have ionizable side chains. Ionizable side chains. So they are going to gain or lose that proton depending on the local environment, right? Depends on the pH of the solution and it depends on the pKa of the side chain. So if we change the pH, we can change the chemistry of the active site. So that's absolutely going to have an effect on our enzyme activity, right? Maybe it's even going to change our tertiary protein structure, right? You change the pH and you might propagate a change through the entire molecule and it might change conformation depending on pH, right? So you can have small localized changes inside of the active site or you can have big changes in the overall protein, right? But what you got to remember is that any enzyme is going to be optimized for the physiological conditions where that enzyme is active. So do y'all know pepsin? Y'all heard of pepsin before? Yes. Where's pepsin? Stomach. It's in your stomach. Yes. Okay, so it's in your stomach. Can you tell me about the pH of your stomach? What's the average pH? Very acidic. Look at this is talking about relative enzyme activity. So how active are each of these enzymes at different pH? This is our peak enzymatic activity at pH 1.6. That is sort of insane for an enzyme to have an activity, high activity at a super low pH. But what's important about pepsin is it's found in the stomach. And so its natural environment is to be in an acidic environment. And so it has evolved to have function at that really low pH. Glucose 6-phosphate, right? This one's in the liver. It's not in an acidic environment. It's in almost a neutral environment, right? 7.8. So this is where it is most active. You just have to look at where that enzyme is functioning and what's the normal pH at that biological location, and that will tell you where your enzyme is going to be most active. Okay, so that's pH. How about temperature? Right? Do you remember when we were talking about polymerase? When we were talking about PCR? And we said that in order to do this, we had to have a specific polymerase. And this specific polymerase was called Taq polymerase. And we called it Taq polymerase because we found it in an organism that was found at geothermal vents. Tell me about the temperature at geothermal vents. Is it high or low? Super hot, very high. So Taq polymerase allows these organisms to survive at high temperatures because these proteins that are enzymes can function at high temperature, right? And we know generally, right, temperature affects our enzyme activity. We know if we increase temperature, we're going to get a decrease in activation energy, right? But you got to think, most proteins, you get up to 80 degrees, you're going to start denaturing those proteins. I mean, they're going to lose their shape. But through evolution, right, Taq polymerase has evolved to maintain its shape at those high temperatures due to, do y'all remember? I taught you this. Why? Why is it stable at 80 degrees Celsius? What's the question? The question is, why is Taq polymerase stable at 80 degrees when most things are stable at body temperature? An excess amount of a certain bond, it's not a bond. But you're on the right track. It's not a bond. It's an interaction. Base stacking interactions. Remember base stacking interactions? We said it was not hydrogen bonds between all the different things. We got base stacking interactions in our DNA, right, that allow that to be really, really stable. And so we have similar non-covalent interactions within a protein that allow it to be that stable as well. So it's really all about non-covalent interactions that stabilize tertiary, quaternary structures in proteins and in DNA, RNA that allow this organism to survive at really high temperatures, right? So this is the last slide, but what I want you to take away from this is that you can change conditions like concentration of your substrates, temperature, pH, right? And we can use this and collect information about our enzymes. So we can understand how an enzyme works and then use this information to understand how, let's say somebody has a disease, and so they have a mutant enzyme. And so we can look at the normal and the mutant enzyme, and we can compare them, and we can possibly determine drug therapeutics to solve whatever problem it is. or we can have, we can study all kind of related enzymes, and we can design possible inhibitors, right? We'll talk more about inhibitors in the next unit, the next section, and say, okay, I need to design an inhibitor that fits in this particular way because of the kinetics of our reaction, right? So enzyme kinetics are super important for understanding enzymes and understanding how environmental conditions control enzymes. And that's all I've got for you, which I wasn't really short because we took a lot of time in the beginning to work out technology. But I will post this recording of the Zoom so that everybody has access to it, whether they were able to attend today or not, right? Yeah, yeah. I figure while we still have power, we might as well enjoy seeing each other. Okay. Any questions? Yes. What you got, Mohammed? When do you think the test? I'm going to try and grade them over the weekend. That's my plan. But I'm also going to post a, yep, hold on. I'm also going to post section 5.3 later. No, we did 5.4. I'm going to post 5.3 later, 5.5 later today. Oh my gosh, if I could talk. 5.5 later today. So be on the lookout, and I'll put it in an announcement on Moodle so y'all know when it goes up. Oh, 7.5. Yes, ma'am. That's what I was trying to get out, 7.5. So our Taq polymerase stability is all about non-covalent interactions that increase stability. So we talked about an increase in stability in the DNA of the same organism, right? And that was all about base stacking interactions. And those are non-covalent, right? And so it's similar interactions that are non-covalent. So they're not permanent new bonds that are being formed, but they're non-covalent interactions that stabilize the protein structure. Does that answer your question, Mohammed? Is there a specific? It's not a bond. It's not a bond. It's not a bond. This is why I really dislike that they named hydrogen bonding, because you could call it electrostatic interactions. You could call it intermolecular forces, intra and intermolecular forces, but you just can't call it a bond. Van der Waals interactions, right? Yeah, that kind of, everything that kind of fits into that category. Just anything that's non-covalent, ionic interactions, stuff like that. All right. So if y'all have any questions, please ask, but y'all are free to go. I hope y'all stay safe and have a great weekend. Why? I would really like to know that, because I would really like to shake the person who did that. I really would. I've always wondered that, and I haven't gone and tried to figure it out. And so I should probably go and do that. I know. Sorry, y'all. Okay, so y'all good to go? Y'all need anything? No problem. No problem. Anytime. All right, y'all stay safe, hunker down, and y'all have a good weekend. Yes, and I'm going to try to have a good grading weekend. I will work on it. And I'm going to post this Zoom as soon as it renders. I'll post it to YouTube. Bye, y'all. Let's see if I can close this.